Hello!
Hi there.
Welcome to YouTube Microbiology Journal Club. We nerd out big about all things small.
My name is Danny. In a previous life, I dropped out for my PhD in microbiology, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicating the public with the tools of biotech.
My name is Faz, and in a previous life I got my PhD in microbiology from Imperial College London, where amongst other things I was making fledgling bacteria glow in the dark. I've also worked as a research and integrity specialist, and these days I'm working as an editor for an academic journal.
Every week, we meet to talk about microbiology, and today it's our deep dive week, long time coming, where we look at each figure in a paper that you guys helped us choose on a Twitter poll.
Yeah, and next week we'll be going back to our news week where we survey a bunch of articles and choose an article for the next deep dive. So make sure to subscribe to satisfy your microbiology curiosity.
You can follow along with the papers that we discuss on either week in our shared Zotero library, linked in the doobly-doo below.
And we want to hear from you, so please use comments or tweet us using the microtwj, well, the Twitter handle that is in that corner down there. No, I always point in the wrong way. But yeah, yeah. So we've got, yeah. This has been a long time coming because we did get slightly delayed because we have this Omicron thing happening. But aside from that, so without further ado, let's introduce the paper that we're going to be delving into, which is going to be called Inhibition of SARS-CoV-2 polymerase by nucleotide analogues from a single molecule perspective. So it's about antivirals, y'all. Today we're looking at SARS-CoV-2 antivirals all the time and every time. And essentially, what I'd like to describe this paper as is almost like an Olympic event to test these
Yes.
different antivirals.
Yeah, absolutely. It seems very high. It's highly driven by the technology rather than the scientific questions in a way. Like they have a very cool technique that can give them resolution into these molecular events, and they're going to use it to test a whole slew of nucleotide analogues, which are a specific type of inhibitor that affects the RNA polymerase of SARS-CoV-2.
So, right. So, firstly, I guess you're wondering what is an RNA polymerase. So, that brings us to a next slide, which shows us like the viral life cycle. So, virus goes into cells, it enters into a specific part, but at some point, an RNA virus needs to make more copies of itself, and that's where the RNA polymerase is
Yep.
important. So, we've
Yeah.
got so
So
it's a very
I was going to say we've
sorry.
talked about various parts. We've talked previously in a deep dive about these. You can see like there's like little membrane vesicles that end up holding all of the double-stranded RNA, and that's because like this virus is an RNA virus, right? SARS-CoV-2, and it's RNA in our bodies is usually used to make proteins. That's the pathway that they go down. But in viruses, they need to make their genomes with RNA. So they have a specific enzyme that makes RNA from RNA. And that's like not something that we would have.
Yeah, it is the hallmark of RNA viruses, and these are set up in a way so they've got like three holes: one for the for a single strand to enter, another one for the nucleotides to enter, and then out the other end comes a double-stranded RNA. And like so, you've got this like fancy computer diagram which has it was I think like a palm domain and a finger domain. So
Yes.
let me see if I
It's all about those
so
it's all those body metaphors for
yeah.
enzymes. I mean, it does help, right? Like I guess as humans, we're very obsessed with our own ourselves, and so if things are made in analogies that hit us on the everyday, right hand rule, left hand rule, then it gives us a better understanding. So yeah, a lot of the ways that people imagine enzymes, especially those ones that grab onto DNA or RNA, is some sort of hand, because you have to slot in that. You can imagine, right? Like in your fist, right? You have fingers, and those fingers kind of like grab onto things, right? They have a palm, right, where things can sit on, right? And then when you kind of make that loose fist, you can see how a protein of some sort would like grab onto a strand. You got a lot of hand
Yeah.
action going on.
Yeah. I'm trying to demonstrate.
Yeah, so I guess that's maybe that. I think that's probably the most important thing. Like you have to think in order for an enzyme to do this action of copying a strand, it has to accept a strand right? It has to somehow read what's going on and then have an opportunity for the building block of that strand of RNA to dock in and then be fused somehow. Like there's a catalytic site, right? Somewhere where the action happens.
Yeah. So this RNA-dependent RNA polymerase is essentially the bad guy in this. All the antivirals want to stop it, and by interfering with how it works, but we don't really know how to figure it out because it's very small. It's very hard. These are very subtle interactions. So what
Yes.
kind of things? How would we figure this out? So Remdesivir, one of the you've probably heard of this antiviral because it's been all over the news. But how does it work?
Yeah, yeah. Like when people have studied remdesivir in the past, they were studying it in the context of just like, does it work, right? Like they put it on cells, right? They try to infect those cells with SARS-CoV-2. We've seen clinical trials, right? The data, the outcome of all that data is like, does it work, right? Is it inhibiting strand elongation, right, of the strand? Is it stopping SARS-CoV-2 from infecting the cells? But what that's not getting at is that how, like, how does it work though? And one of the first, I guess one of the first clues that sort of came out from this is that it could be a chain terminating, right? Like chain termination. That was one of the biochemical outcomes that was looked at. So at first they incubate some template strands of RNA with the preferred with the polymerase, and then they see, and when they add in bits of remdesivir, they find, oh, they're getting they don't get the full length that they would normally get. It stops at some point. So they have shorter products in the the condition where they use remdesivir, and they have longer products in where they don't have remdesivir. And what was interesting too for them is that RNA polymerase, the one in SARS-CoV-2, it has a proofreading function, and that has been thought because SARS coronaviruses have really long genomes, longer than you would expect, I guess, from a piece of RNA. Like when we make RNA in our bodies, they're just for every protein, but for these viruses, like it holds all of the proteins they'll ever make. So it has a rather large genome, and consequently, they think this exonuclease function of the RNA-dependent RNA polymerase is important. And you can see that if you knock out that exonuclease function, remdesivir does better at stopping stopping those viruses from entering cells. So this was like kind of the first indication that maybe remdesivir could overcome, right, this particular mechanism that SARS-CoV-2 uses to keep its long RNA genome intact.
Yep, and so how can we measure it? So there are a variety of tweezers out there. So I've heard of sonic tweezers, optical tweezers, but today we're looking at magnetic tweezers.
Yeah.
Where we're looking at how, using a magnetic field to kind of stretch a DNA strand, well, not DNA, but an RNA strand. And the thing is, there's a unique thing about about DNA strands, or RNA strands. When they twist, they become slightly shorter. And when that happens, that means, and the tweezers are a way of like measuring that displacement very easily, well, not easily, but very sensitively.
Yeah, I mean, I think anything at this scale, right? We're talking about single molecules of DNA, right? Kind of twisting around. So like that difference in distance that's made is like it's on the scale of single base pairs in space. These are not big. Yeah, they're not big effects that we're seeing. So we have to have very sensitive equipment to be able to see it.
Yeah, and so like first, so you got this magnet that is used to stretch out this strand of RNA, and one of the things that happens, like, so imagine if you got a balloon on a string and you twist that string down, the balloon is going to get closer to you. Same thing happens with DNA or RNA when that gets twisted up, and the thing that makes it twisted up is if it forms two strands.
Yes.
So
yeah, basically inherent in the chemistry of nucleic acids, right, is this propensity to, first of all, find their base pair, right? Like that's something they like to do. But then also their most, like, I guess, energetically favorable conformation is to sit in that double helix.
Yeah, and let's see if I can.
Yeah,
so yeah,
so all tweezers will then work by this principle of like basically like some way to apply very small, like this is like piconewtons, I believe is the type of forces that we'll be talking about, tiny amounts of force to a strand of DNA. Because you imagine any more, you could apply enough DNA to just rip it.
Yeah.
Right? These are physical objects. They're tiny physical objects, but very, yeah. And so they have a chemical method to adhere both ends of that strand of DNA or RNA, where just any nucleic acid falls into this the set of techniques that we'll talk about today. Adhere them to beads, plastic or metal, whatever, some sort of bead. And that bead could be fixed onto a surface, or that bead could be pulled by your tweezer of choice.
So in this case, using the RNA polymerase, essentially turning a single strand into a double strand, pulls out the ssDNA into a dsDNA. So what effectively happening in this graph, you've got like ssDNA which can stretch a lot longer, and then dsDNA which doesn't. So effectively like changing it, moving it from one graph to the other, and then shrinking it down.
Yeah. So I guess like as the RNA polymerase is making that second strand, right? Then the double-stranded DNA is going to begin to contract, and that should move whatever the bead is on the one end. And because you have these tweezers that are applying a force, it'll keep it like extended, right? Like there's no effects that are gonna like collapse it with outside of the force of the polymerase moving across it. But that doesn't get at this idea of how do you measure how much contraction is being done. This is only talking about the way that the force is being used to extend that strand of DNA. So yeah, what you were saying, yeah, we're moving between these two. It is possible, right, to pull DNA to pull DNA past to its stretching point, if you will, right? Like that. You can see I think this is single-stranded DNA, right? Single-stranded DNA, that force diagram, you can extend it quite a bit, I guess. It seems weird, but it's like more flexible to extend DNA.
Yeah, but at 60 piconewtons, you suddenly see the double-stranded DNA suddenly behaves like single-stranded DNA. That's at the point where it unwinds, effectively. So
yeah. No, it's like a lot of force goes into it. Like you need. Yeah, all of a sudden, like it'll just like pop, I guess, right? You could pop the double-stranded DNA back to its back to being stretched out. But that transition, having the force measurement for that transition is is part of the measurement here, right? And why this technique is so useful for studying these small molecular dynamics.
So now we've got like the setup they use. So how do you measure this? They've got so what they effectively have is a microscope slide with all these little strands of DNA attached to them and the little magnetic beads. And so in part B, in B you can kind of see all these little dots. Those dots are all magnetic beads. And this is how subtle they get. They don't measure like the way they measure distance is actually how out of focus they get. So they've got like a specific. So I don't know if you're a glasses wearer and you take off your glasses and things are out of focus. It's like using that, but as a way to gauge distance. So effectively, like the more blurry the more blurry these beads look, the farther away they're getting from the focal point.
Yes.
And so you can, once you know a certain level of blurriness, you can kind of guess how far away the bead is getting, and they use a computer to calculate this.
They basically, it's like they have a calibration, right, where they take a bead and they move it through the focal planes, right, and they're like, okay, it looks like this is how it looks as it moves all the way through, and so then they make a huge lookup table of all the different images, right, of that bead moving through the. People usually call this the Z plane or the Z plane, right? It's the distance from the top to the bottom when you're looking at a microscope slide, as opposed to like up and down or left and right. So like
for viewers at home, it's like when you're going to 3D cinema, you're up, right, and the bit that's poking out at you, that's the Z plane.
Right, and so that's what they're looking at. That like bit that's poking out into the focal plane of of the microscope, that is going to be more, or that will be in a different focus levels, right, depending on how close or far it is from you. And yeah, they make a big lookup table, and that becomes calibrated to distance. So at the end, what we're going to get an output of is a distance graph, right? How far is the SARS-CoV-2 moving, or how far is this bead moving, right, as the double strand is being made.
So we've got, so yeah, what we have is like what's called a Chymograph, which essentially the bottom axis is time, and the top axis is the movement position. So it's fairly easy to figure out. So if something's moving very fast, then the line will be very diagonal and very quick, and if it's very slow, then it'll slope. So like I guess now everyone's familiar with this basically because outbreak graphs like for Omicron and other things are basically like this. So
yeah, I mean, it is a rate, right? Like I mean, also if you've taken high school physics, if you can recall back to high school physics, this is essentially that sort of diagram, right? Like these are diagrams of things moving over time. There's an you can see acceleration, right? You can see a velocity. You can see when that velocity peters out, right? Yeah, these are the types of things that they'll be able to study. And remember, they have a huge microscope slide full of as many beads as they can adhere to it. So they're measuring from across like a huge field of these objects. And that's a very powerful technique because, you want to average. Biology is not 100% in every scenario, right? There's variability all over the place. And capturing, right, a large statistical set of single events from a molecule, that is teaching us so much about the behavior of that particular molecule. So in this case, right, we're talking about RNA-dependent RNA polymerase, and then they're going to sort of mess it up a little bit and see like how it's being messed up by the various drugs that they're adding.
Yeah. So this is the method by which you're going to measure how successful our antivirals are, because the idea here is whatever can slow down the RNA polymerase or mess with it the most.
Right. And I think specifically how, right? Because again, like we know that it does at some level mess up these polymerases because there's all these other studies about doing it in cells, right? And this and that. But in this model, like they're focusing very much on the molecule RNA polymerase, and it'll have a characteristic. We'll see it probably a little bit later. There is a characteristic way it works in this condition, and then when you add in these nucleotide analogues, it'll be messed up in different ways, and they interpret that. There's a lot of interpretation of these kind of
stuff. Yeah. Which kind of like brings us to the next slide, which is about the behavior of this RNA polymerase, because it doesn't.
Right.
So yeah, it doesn't work the same. Or there's like there's a couple of different pathways that allow it to work.
Yeah. So
we've got.
So this is like something from previous studies, right? This lab has worked with these tweezers before, and they just did like the baseline, and they'll do the baseline again in this paper. But from their initial study with the baseline of RNA polymerase is after measuring its behavior, right, extending things over many different conditions, right? They had just like many replicates of it. They just they binned the behaviors into different categories, right, based on the slope of the line of the chymographs that they're seeing, right? So, some are moving fast, some are moving slow, some are pausing, right? Some are going backwards. They can see that as well, right? If the double helix is being unwound as the protein moves backwards, they'll see distance increase again, or I guess decrease because it's like they're being floated back to you. And so so they build this statistical model, right? Essentially by taking the frequencies of all of those different conditions, like fast slope, slow slope, backtracking, and they tie them together with probabilities, again, based on the distribution of all the different events that they've seen. And don't worry, we will basically be talking about this over and over again for every single nucleotide analog that they try, because they're always fitting their data back into this model, right? That says that describes 3 different speeds in which the RNA polymerase could be moving, right? There could be like slow nucleotide addition, right? That's SNA. Via SNA is very slow nucleotide addition. And NAB is, just nucleotide addition. I don't know the
Burst.
burst. Oh,
So it's
burst. Yes.
yeah,
Yeah.
nucleotide. So yeah, there's one that's very fast. So burst is like the first gear,
Hmm.
and slow is like no wait. I got that wrong because I'm bad at driving. But burst is very fast. Slow is slow, and very slow is like very slow, and backtrack is essentially like the reverse gear.
Right.
First gear is not the fastest. I just remembered that.
That I don't drive at all, so you're on your own in creating that analogy.
Yeah. So
Yeah, but so yeah, so you
yes.
can see like these are basically zooming in, right, into particular instances where they would make the call, right, that this is a pause, right, or this is I guess in this case it's all pause probabilities here. So like there's both, right? You can characterize once you characterize like once you characterize some effect, right, in terms of like how fast something is moving, then you can say like what's the probability across all the different single molecules that we've looked at, right, that we will see, right, polymerases in this behavior mode. And of course, that could be dependent on many things. These are enzymes, right? These polymerases are enzymes. So it matters like how much substrate they have. I don't know if people recall, this is like is it a high school? Maybe it's early university, college level sort of like biochemistry, right? But like enzymes and have behavior that's dependent on their substrate, right? Like when you crank up the amount of substrate, sometimes that will speed up the enzyme until it's working at its optimal level, and then it gets saturated with everything that it needs and it'll slow down. So the same way, right, essentially we're studying this enzyme in 4 different modes of it working, right, based on statistical behavior. And yeah, we can slot in a full range of nucleotides to see if that has an effect. And then also, another thing that could affect this enzyme is one part of its substrate is the mRNA, or not mRNA, but the
Yeah.
RNA strand. And so there's also an idea that the tension on that strand could also change the way that that enzyme is moving. And so they also have the ability, because they have these magnets, by moving that magnet closer or further away, they're able to change the force applied to those strands of DNA. So they can also see force dependency here. All very complicated, actually. Like I don't think I could walk people through any more detail than that. And I'm sure that's
Yeah.
already maybe too much.
But I mean, I guess like simplify it is like there are like 4 different behaviors that this enzyme can experience. So like 4 different speeds, and in
Yep.
order, so that's why it's kind of difficult to figure out the kind. So I think we'll explain it a bit more. But
Yep.
the entire point of the probability is to break each of these things down, because if you did
Yeah.
the average speed, it wouldn't tell you much about it, because all these enzymes are behaving slightly differently. So
Exactly.
we'll
Yeah.
explain that a bit more
Yeah.
in a bit.
So yeah,
But
this is we're diving right into the setup that they use, a setup that has been used in previous papers as well. And so that involves, as you saw before, adhering all those different strands to a bead. They're actually made as a hairpin, and I think that's just for like storage reasons. So that's figure B here in this on the right-hand side. That's how they create their test molecule. I guess that's like the most stable version of it. And then you can see at the top they have biotin, and at the bottom they have digoxogen. I don't know how to say that.
Digoxygenin or
Dioxygenin. Yeah. So those are.
digoxygen. There's a G in the middle that trips me up. There's too many G's.
I don't think I've really said it out loud that much. But those are both sticky types of biological molecules. So the biotin at the top allows them to stick the molecule to a bead, and the dig at the bottom, the dig at the bottom, allows it to stick to the glass. And so those hairpins get adhered, right, to the flow cell, the experimental model, where they have the microscope set on, where they have the pictures being taken. And then when they apply that magnet force, right, they can apply just enough force. They find the perfect amount. I think they say that that's like they do their experiments at 35 piconewtons, right? That's at the top of this graph, where they pull and they can see that the hairpin pops. So they know
Right,
that they
yeah.
have a straight right now they've straightened out their strand, and then they add in recombinantly produced. So that's figure A, right? This is the proteins they make: NSP7, NSP8, NSP12. Those are the components of the RNA-dependent RNA polymerase. So this is not even just one enzyme, right? Or it is one enzyme, I guess, but it's like made of a composite of a bunch of different proteins.
Yeah.
And they add it all in, and then they let the reaction, and then they put nucleotides over because they need nucleotides to grow the strand, and that's the experimental setup.
Yeah, and so we get trace like this that shows you the speed, and it's
So
here you can kind of
every line is one molecule here, right?
yeah.
Yeah.
And you can see that some molecules are going at different speeds, and sometimes you can see where it stops, like the actual like growth of the strand stops completely,
Absolutely.
and then it can restart it again, and so there are also so how do we like classify like how slow it is, and
Yeah.
like, because if you took
I was
yeah.
gonna say, zooming in, like looking at that insert is also really helpful, to me at least it was, to visualizing what's going on, because like that is the level of resolution they have, right? Like they can see pauses, like you can see in some of those traces, they kind of like, oh, they're slowed down a little, and then they went back up, right? Like that is the resolution of, right? That's the resolution that they're able to see with this model. Like some of that might be noise, right? That's like, it's like, but in some ways that's all that biology is, right? Like it's like that noisy behavior being focused down like a certain direction. And anyways, so like that's what we're seeing. Like those small fluctuations in the lines in the zoomed in insert, those moments are being classified, right, as being pauses or burst additions.
Yeah, that's why they're trying to classify the behavior of the polymerase rather than just taking the average speed, because
Yes.
that doesn't tell us. Because average speed would tell us something about how fast it's going, but it doesn't tell us about the behavior. And what we're interested in is how antivirals will change the behavior
Yeah.
of this enzyme.
Yeah. So
And
like
so that's why we.
yeah, that's a really great way of saying it. And I'll say like, so if you were to average it, right, like then you could just look at the zoomed out ones. You say, oh, look, it's a line. It goes up, and then some of them stop and some of them don't. But when you zoom in, you get this like resolution, right, that can tell us more. Especially because it fits into this model that they have, right, of the nucleotide burst, the very slow, and the slow.
Yeah, and in order to like figure out which kind of what the behavior is, I think they use this thing called dwell time, where they take 10 seconds, measure the speed in that 10 seconds, and then like they basically break up the curve into that
Yep.
to figure out, because the thing is these behaviors can be shown by the same like RNA polymerase during a run, and we'd have to be able to figure that out. So they use a dwell time to figure out how quickly it's going,
Right.
and then
Right.
based on those chunks, like not just looking at like the RNA polymerase on their own, but also looking the amount on average how much of this behavior is being expressed.
Yes.
And so
Yes.
that's
Yeah, no, that I think that's a great description. And one thing that I'll add to that is this is in my mind, like from the microbiology world, this is very similar when people look at when people are doing data analysis on sort of those large sequencing sets, right? This is a concept of binning, right? Like they're binning, they choose a window that they scan across, and then they bin those events into like these are the bins, the pos1, pos2, the backtrack, and the nucleotide addition rate. And yeah, that's based on how much time that they're dwelling in a certain spot.
yeah. So imagine like this is like a crowd of people, and you can divide out like the fast walkers who are in green, and or people, well, not even fast walkers, but the people who are walking fast,
Yeah.
because sometimes a fast walker gets stuck behind a slow walker,
Yes.
and then you have to like
Yes.
parcel out that behavior.
Yeah.
So you've got like the amount of fast walking that's happening, which is in the far like left of this graph. Then you got various like slow walking like degrees, and then you got people who are just backtracking or just mulling around in place.
I love it. That's great. I think that's a good way to move on.
And so that's what these kind of curves present. So we're going to be seeing a couple of those. Also, like this is an interesting one. So now looking at the average replication time, which is what we talked about a little bit, and also the product length at the end of it. So
Yes. Yeah, so this is more of an aggregate style, right, measurements, but they do give them as these violin plots, right, which shows you that the distribution when you average is like kind of wide across everything. And the changes that they're looking at, I think, are the medians, right? The lines in these violin plots are the medians. And this is just the baseline, so they're doing it across two temperatures. They already know, right, that when they do them at a higher temperature, things go faster. There's more. That is a general rule in biology, right? Something that comes from the chemical world, right? A chemical rule saying that the rates of reaction are faster, right? Adding more energy into a system. It holds true here, right, in aggregate for biology as well.
Yep, 37 degrees is human body temperature as well. So I think like another thing is like obviously like when you get too much energy, then things start bashing to each other and breaking. So this is at the kind of right space. They're trying to like mimic like the natural situation, also an artificial situation that's somewhat cooler.
Yes. Yeah, so I think most of their experiments they'll do at 25 degrees Celsius actually, for a reason I could not determine, because they say when they raise the temperature at 37, they see better resolution for some subset of events. So I think it's a resolution thing at different temperatures.
Yeah.
Oh, okay, go ahead.
It's also, I think, like partly like the behavior of they want to like, they don't just want to like see how it reacts at like 37. They want to see how it behaves, and slowing it down, I think, allows them to see like get longer traces and get more information out. So like extending that time, you're kind of like running things in slow motion so you can pick out those smaller events. And also, like, if you're looking at these violin plots, there is a lot more variation at 37 degrees, which is natural. Lots of things vary, but if you want to like really pin down like an effect, then you want to have little variance, and you want to be able to like pull apart and see what's happening.
Right. Yeah, that's great.
Yeah, and then we're looking at dwell time again. So you've got this probability density graph, and like essentially like the further to the left it goes, the more quicker the RNA polymerases are working. And you can sort of see like that is a case for at 37 degrees. At 25 degrees, it's shifted slightly to the other end, but the actual like backtracking time hasn't changed.
Yeah, like it looks kind of the same. It's just being pushed out a little, translocated to the right a little. And you can see the difference in the rates, right, that they measure in H. So then you can just take, you can integrate? No, you can take the slopes, right, in these different areas and come to a rate of incorporation.
Yeah, and what they find is like at 37, there's more nucleotides like added, but there's also more polymerases that are in pause 1 or pause 2. And I think they want to limit that so that they can see like a bigger effect. If your antiviral is having an effect, you want to be able to see that effect. So then they've got like some data here for some of the replicates. So you can see like normal polymerases. Sometimes they like cut off early, but slow is very like slow.
Yeah. Yeah. You can see a difference, right? Like, and this is going to be important because we want to be able to see how the nucleotide analogues are inhibiting things.
Right. And so you can see like that there's changes. So you've got your kcat, which is your rate. You got your probability. So what's the probability of something being in pause 1 and pause 2? And then what's the probability of things being in backtrack or not? And kind of like mirrors what we've seen before. So again, this is looking at 25, 37.
Yeah.
So 37 has more in pause 1 and pause 2, but backtracking seems about the same.
Yeah.
And here we have our contenders, nucleotide analogues.
Yeah, they look like nucleotides, and so an enzyme might grab onto them, thinking like, oh, let's add this into the chain, but they're usually missing or have odd additions to them that might cause some problems if they do end up getting incorporated, or maybe they can't get incorporated at all because they're missing some of the groups that would be required for that.
Yeah, so they sort of match a shape enough, so they look like food, but when you bite into them, they're made of plastic. That's kind of like what Yeah, I
Yeah.
don't know if you've ever seen those like delicious menu items in Chinese shops where they put out these
Yes.
plastic versions of their food, and you're like, that looks so tasty, and it's just plastic, and it's just.
Yeah.
So the first one we're looking at is the 3D ATP, which is sort of like ATP, but it's missing hydroxyl group, so it's kind of like a different shape.
Yes, and
And
that's
that,
really so that is really important. That hydroxyl group is used in forming the glycosidic or the phosphodiester bond between the phosphate and that OH. That's the zoom in here for the DNA polymerase. This is also true in RNA, right? And yeah, so without it, the concept would be that polymerase couldn't do this chemistry, and so as soon as it incorporated a DATP, that would be it. That strand is now just dead in the water because all it has is, well, it doesn't have the right chemistry to continue elongating the chain.
yeah, like the hydroxyl group is like, I don't know, let me think, like a Lego piece that, and
Yep.
like, except now like you're trying to connect a Lego piece with one of those smooth pieces and it doesn't work.
Yes.
It's like that,
Yep.
like,
Uh-huh, uh-huh. That's really good. I like it.
and yeah, it ends the chain. So the
Hmm.
question is, how does this fare in this model? So we're adding 3D ATP to this experimental setup to demonstrate that what's it going to do to the RNA-dependent polymerase? And so,
Hmm. Yeah, I guess
uh, uh,
unsurprisingly, there are all these stopped traces, right? Like things where the traces were dead in the water after a certain point.
yeah, so it terminates it. So what that we expect for that is that the maximum product length gets reduced over
Hmm.
time, and sometimes you get other polymerases reattaching to the molecule trying to do something on it. But yeah, effectively, like what you see is like the product length is just much lower.
Yes, yes. Yeah. So, oh, sorry. D though is sorry, we go back to D, and so like the product length is much lower, and then in D they lower the amount of ATP. So now like this is a case where they have a lot of chain terminators, right, relative to the stuff
Right.
that's going to let extension happen, and you can see that all the traces have been severely depressed now, right, because it's more likely that a polymerase is going to find one of the chain terminators and put it in there. And so you'll see like all the product lengths go down as you increase the stoichiometry, right? The concentration of DATP versus ATP.
Yeah, I think that's an important point. They'll be manipulating that a lot over this experiment just to figure out like which one of these is doing better at getting into the RNA polymerase.
Yeah.
Which brings us to next slide, which is competition. So a good so how easy can this like fool an RNA polymerase
Hmm.
into taking up? So we've got this thing called a competition assay, which does the math for that. So
Yes.
um, I'm not gonna lie. I didn't really understand this figure particularly well.
Okay. Yeah. I mean, I think the way that I picked it up was, yeah, what you're saying. There's a probability, right? There's both a probability and a rate in which that the natural nucleotide can be incorporated versus the terminating nucleotide. So again, this is just a sort of a way of understanding, right, the knobs that we have to control. Yeah, the knobs that we have to control in order to derive some sort of like constant or something like some single number or that we can use to compare things towards each other. And yeah, that's exactly what we saw in the last one, right? Like if we turn up the concentration, like the probability of an event happening, right, is based on concentration, right? If you had more of the terminating object, right, more of that, in this case, T or N, right, the elongating or the terminating, um, that sort of works in concert, right, with the constant that we identify there for when it would transfer to either of those states. So just a way of
Yeah,
representing things. But right, we say here like from this whole model, they can do some math. Like they had many equations in this paper that I did not uh, clip out to show you guys because like I couldn't talk any more about them. But all that to say is that like they do have a single number called the effective incorporation rate, which is represented by gamma, that they will sometimes calculate from using a model like this. They'll sometimes calculate to determine how many nucleotides on average can be added to a chain for a given concentration, right? A given concentration setup until a terminator is added. Right, and so a smaller number of gamma, right, would mean that that is a very effective terminator because, like, for whatever reason, whether that be the power of the termination event or maybe just like the affinity between the terminator and the enzyme and the polymerase, yeah, low numbers will mean that it's more effective at terminating. Higher numbers mean that it's less effective.
Yeah, I think that is an important point, and yeah, so because like firstly you want to figure out how often like this will run into the polymerase, and also like because also like these are different shapes, so they might have more trouble getting to where they needed. So
Yep.
you want to be able to know like what that probability of it actually being incorporated. Because if you give something that's great at like binding to the DNA strand, but they can't actually get into the polymerase, and it doesn't necessarily work. So
Absolutely.
that's an interesting thing to figure out. So here they're looking at the gamma in the condition where there's 500 micromolar ATP versus
Yeah, so they have a whole
50 micromolar.
they're changing up the ratio, right? Because to
Right.
get at this data, they have to vary some component, right? So that they can find like the underlying relationship, right? So they're going to vary the amount of DATP versus ATP, or it's a ratio that they're showing on the x-axis, right? And
I see.
yeah, and from that, right, they plug that into, again, I didn't show whatever equations they're using because I'm not going to like be able to explain it that detail, but you plug it into essentially an equation that says, right, like what would the constant be, right, if we varied these things, solve for that constant, solve for this gamma or whatever, and that's what they kind of output these gammas. So at the 500 micromolar ATP concentration, right, they get a gamma of 780, but at the 50, when they lower the amount of ATP, they also still get 770. So that just means that doesn't matter how much there is a good affinity between right, DATP and the polymerase. It's not being affected that much by removing ATP from the scenario.
Right, I see. So it's so there is the effect that sometimes you can block off an enzymatic inhibitor by having something that's very similar. So
Yeah.
so if like ATP is much better at binding to the RNA polymerase, it can actually block your
Exactly.
drug.
Yeah, exactly. Exactly.
So what this establishes is that the concentration of ATP doesn't have that much of an actual effect. It's all dependent on the concentration of the 3D-DATP, which
Yes.
is our inhibitor.
Yes.
So
yeah, at least that's
right,
least
okay,
that's my reading. So
Right.
let us know in the comments as always if that seems totally wrong to you guys.
Yeah, it's quite hard to like compare these two graphs because they are on different scales. So one of them is saying like a 20-to-1 concentration is pretty good.
Yeah.
And so yeah, I'm trying to like link these up because I think like that is probably the two and that is two. Am I? Yeah, I'm. But
Yeah, like
but yeah,
it's basically in each of these, like the total amount of ATPs is definitely changing as well, right? Because they fix one concentration and they vary the other. I don't know. That's that's all I can say about this. I don't know if I can. But we're gonna
okay.
but we're gonna see this over and over again, right? Like this type of experimental design is essentially the hammer that they're going to be applying to every different nucleotide analog to tease out, right, what the characteristics are. Yeah, so we just zoomed out to see two more graphs, which is the replication time, right? So when they add more or less, right, of more or less ATP, right, and a varying stoichiometry of the terminator, does it have any effect on the time that it takes? So then just the overall time that it takes for things to happen. And you can see that there is no big effect, right, at the 500 micromolar ATP. Everything seems pretty constant. But once you get those really dramatic changes in product length, then you do see some effect on the time. But the authors are saying that that's driven by the fact that it takes less time to like make a 195 nucleotide product than it does anything else. So there's nothing unexpected in this data either.
Right. Okay. So next one, we're looking at Remdesivir. So here we've got our spot difference where you got like a cyanide group, I think that.
Yes. That basically is like in the way, right? Like that's not present
Yeah.
in regular ATP. So this is probably going to gum up the works in some ways. And it is actually, it was previously discovered, right, or proposed that that is the case. They did some more detailed biochemistry to come to a model, which they have a wonderful video of.
Yeah, which we're gonna sit down and watch.
Yeah.
Let's watch this. So now we're zooming into the RNA polymerase, and what we're seeing is that that's the DNA strand, and you got your remdesivir. It's getting added onto it.
RNA strand. Yep.
But then it continues. Yep, RNA strand. Sorry. Yep, and then you've got like other nucleotides being added onto it. So it doesn't turn it immediately.
Right.
So it's still in there. So what will happen now? So it looks like everything's going normal, right? But then we have a clash.
Yeah.
So yeah, it gums up the works.
Yeah, so like this is the way the enzyme works. It has a single catalytic site, and it's like moving that strand of RNA through the body, right, of the palm, if you will, right, of this enzyme, and things are being acted on a single chemistry site. That's where the elongation is happening. Remdesivir, it's not missing the molecule, right, to do that chemistry, right? It can still do the elongation chemistry, and so it will go along, and things will be added to the OH, right? The phosphodiester bonds are being made, but that extra bulky group, as the strand of mRNA is moving through the RNA-dependent RNA polymerase, there is a part that's a bit, the geometry wasn't made for some bulky object like that to be there, right? And so it gets stuck. And then that's thought, so the thought would be that that is what's creating like a more pausing, right? And then thus then it would terminate, because things would just, all these enzymes would be stuck at this point, and they would just disassemble at some level, and then if they reassembled, they'd also still be stuck. Yeah.
Yeah. So that brings us to the next graph, which looks at remdesivir, and what you see is that there are a lot of terminations, and and so again we've seen, now we kind of understand the measurements we're looking at. So we've got looking at, say, position. So that kind of graph, you can see lots more of those, like these long pauses happening where the RNA, the polymerase just gets stuck and can't really get further. So I imagine what happens is its choice is either to disengage from the strand entirely and wait for another RNA polymerase to run into the same problems, or just slow down and
Yeah, the replication time is going way up. Like at some level, it does complete some, right? Like the product lengths, it gets wider at in figure B, right? At the most extreme end, 0.6 remdesivir to ATP. The violin plot has gotten a lot fatter, right? Meaning that there are many instances where things are being terminated, but on average, the median is not that much decreased. It is, but not like dramatically so. It is finding a way to make those strands in somehow, but largely it's getting terminated a little bit early. But the time, I feel like, is quite dramatic, right? Like you can clearly see that the time is going up across across all of these as you increase the amount of remdesivir, which makes sense, right? Because the more remdesivir, the more of these pausing events you get, and we're not right. There may be ways for these molecules, the RNA-dependent RNA polymerase, to edit out this, right? Like maybe they can reassemble after the fact, and that allows for extension, or maybe they somehow excise that strand, right? That could be like a proofreading ability. And so so again, we don't know exactly from this data, but it's giving us more. It's just giving us another observation that then we can start aligning with the different models that we think this is actually working, how we think this is actually working.
Yeah, another thing I'd like to call out is at the bottom, the ratio of remdesivir, it actually seems to be working before it gets to a 1-to-1 ratio with ATPs, which is actually really active. So that means that essentially it works at, so at 0.6, where there's 0.6 per 1 molecule of ATP of remdesivir, it's a dramatic. So it's gone from 25 seconds to 350 seconds. So in ping terms, that is a really bad connection for Remdesivir. It is clipping through the wall at this point with adding Remdesivir.
Yeah.
And so, like, we've got a few example traces to show, like, this kind of backtracking activity that is caused by Remdesivir because it gums up the work. So essentially, what you're seeing is the position of the RDRP goes up and down because it's like it's just staying in place while it kind of like chokes a little on Remdesivir, I think.
Yeah. Well, I mean, it's thought that like backtracking ability is like this is a thermodynamic right possibility, right? Essentially, like the ratchet is trying to move forward, but then something blocks it. So then at a certain if being blocked for so long happens, then maybe it's actually favorable for it to just keep going backwards for a while, right? Maybe to find a problem. Like this is, in some ways, backtracking is the basis of proofreading. That like you can't have proofreading if you don't have backtracking, because you imagine like the catalytic site of the proofreading like exonuclease, like it has to move along the strand if there's a problem. So this blockage, in some ways, the behavior that we're seeing from the SARS-CoV-2 polymerase, I would say that this is probably very adaptive for this RNA, for the RNA polymerase, and not all enzymes would do this. I would say, right? Like this could be something that like is unique to the SARS-CoV-2. I'm again, I don't know if it is, but like not all enzymes would do this because maybe this one is more likely to backtrack more in certain cases because it has that proofreading capability. If it didn't have it, then it would just dissociate and not backtrack, right? So like those are the, I don't know, that's like just some of the stuff that I was thinking about. Again, not like a super well-versed in the literature of RNA polymerase kinetics, but again, this is an amazing view into that into that world, and probably better interpreters than I will have more concise things to say about seeing these traces.
Yeah, and so so now we've got these dwell time graphs which give us a probability trace based on like how. So
and you can really see that is like I think the darkest line is the no Remdesivir, right? And as they added Remdesivir, the line gets lighter, and you really do get a big shift there in the probabilities of being in the backtrack. Or is it the opposite?
I think it's the opposite way.
Oh yeah, it's the opposite. So yeah, because you're getting more backtrack. Yeah, more probability of being in the longer dwell times.
Yeah, so Remdesivir basically slows it down. It causes the RDRP to just, the polymerase to just choke a little and stay in place, and that causes a shift in overall dwell times. So yeah, now we've got like applying that model to estimate the rates
of
being in these different like pauses and nucleotide.
Yeah, essentially, these types of graphs are like summaries of seeing that dwell time graph, right? Because from those dwell times, they pull out these probabilities and plot them against the Remdesivir concentrations.
Yeah. So like Remdesivir, basically, it seems to increase the backtrack probability by a lot. That seems to be like the main effect here, and everything else seems to be going up slightly. But and that's really interesting. Because again, you wouldn't get this if you just looked at the time. You wouldn't understand the behavior of the polymerase if you didn't have this kind of setup to look at the individual behaviors.
Yes, yeah. Like all these things would be like lost in the average. This is the whole this is like single molecule biochemistry is like on the coattails. Same conceptual idea of what gives single cell biology such an amazing ability is that like instead of, I think I've seen this in the single cell like marketing literature. It's like it's like biology is really varied, so it's like all these different fruits. If you're just blending everything together and taking the average of that, right? Yeah, you could tell if the smoothie was like more banana-y than another, right? Or more pineapple-y, but you wouldn't really know like how many bananas, how many pineapples, and what about the blueberries that were in there, right? Like,
yeah,
doing things at the resolution of single molecule. They're using that marketing material for single cells, but doing it at the resolution of single molecule gives you that same insight, right? It doesn't blend everything together, so you just see a single average.
Yeah, exactly. And so now we've got like
Oh, they did it with the
Oh
classic
yeah,
SARS. Yeah,
SARS-CoV-1. Yeah,
SARS-CoV-1.
original recipe SARS to see
Uh-huh.
where it has a similar effect on the behavior in terms of
Yep.
inducing backtrack. So again, it
Mm-hmm.
replicates similar to what we've seen in previous graphs.
Mm-hmm.
Not quite as dramatic as with SARS-CoV-2, but it's
Mm-hmm.
still there.
Yeah, because
So
here they
that's
see a
good to
big
know.
increase in pos 2 as well.
Yeah,
Mm-hmm.
yeah, So yeah, it's so moving on to the next one, which is ratio of uh, adding just more nucleotide to the. So rather than just like so, so yeah,
Yeah, this is like in the first two graphs they also right like they did the 500 versus the 50 right like this is now in the 50 condition.
right.
Yeah, and so these are the same like summary graphs right product length replication time and probability density but this time there's even less ATP right so there's more more proportionally more remdesivir in these conditions.
Yep, and again we're seeing that shift
Hmm.
to towards
Yep.
backtracking. Uh,
It's still working in this condition.
yep, and now what we have here, we've got ratio of
Same stuff. This is the same.
same stuff.
This is the same one.
Yes.
This is now we just can see the two summary graphs. Maybe if that was easier for people to see, like instead of having to read it off the violin plots, you can see rate and probability. Yeah, written just in bar. Yeah, you can really see backtrack. Right, the backtrack condition it jumps up a lot from.
Yeah.
Yeah.
And yeah, so that's really good at like slowing down the virus at replicating.
Hmm.
Yeah, and now they're looking at temperature again to looking at
Yeah.
how that might have an effect.
This was in the supplement. Like they said that the reason they used temperature was because helps them better distinguish between the pauses. For
Mm.
some reason, again, this is knowledge that they gained from setting up this experiment earlier, and yeah, they're doing they're repeating the same experiments to try to see if they'll see a difference in the pauses, I guess, at this particular temperature. And they do sort of see it in pause 2, but it's still not that much. The backtrack signal is still the clearest, is what they're saying. They see still a clear backtrack signal at this temperature.
Yeah, I think this is important because this is the temperature that SARS-CoV-2 will experience when inside the body. So this will give us
Hmm.
an idea of how applicable the results we've seen previously are to the real situation. And it
Sure.
does seem
Yeah.
like that. What they found the backtrack is important. And they do still see some stuff with pause 1 and pause 2, but it
Hmm.
seems to
Hmm.
be going in a different direction. So yeah, this
Yeah, so
is
the
interesting.
rates, I think what's important to remember when we see rates for the pauses, like what's the rate for a pause, right? It's the rate, I think what they're doing exit rates. So it's like,
Mm.
yeah, how often does it exit, I guess.
Oh right, yeah, because there is like sometimes when it can come. So there's a big line, and then they can come back afterwards. So they're trying to look at how easily it can get into and out of a pause.
Yes, into and out of a pause. Hmm. Hmm.
Yeah, it's like when you're halfway through a sentence and need to get out of it somehow, and you get to the end of it.
Yeah, I feel like that can be sometimes how we talk about these things.
Yeah, like when you
um,
get into a Zoom call and you hear someone else want to say something, and you pause, and then they pause, so you realize and everyone wants to hear each other. But because everyone wants to hear each other, we all stay completely silent.
And then here is another control in a way, right? Just to make sure that the conditions that they're doing this experiment at are not the conditions that lead to this phenomena. And so, we said before, they were typically using 35 piconanometers, piconewtons, oh my gosh, piconewtons as the force to pull and pop that hairpin. But they can go as low as 25, and I think they still get the pop. But it holds less tension on that strand, but they still
Mm.
see these effects, right? When you add more remdesivir, you get that increase in backtrack probability. Mhm.
Yep. Now we've got our next contender, which is the great name T1106TP, which is a more stable version of favipiravir T705, and this mimics both, I think, cytosine and uracil, right? Because
Yeah.
it binds to ATP and GTP.
Hmm. Hmm.
So
But it has like a part of where it would normally make hydrogen bonds with the complementary base pair. It has a bulkier sort of group there that would probably cause some issues.
yeah, this is interesting because with this one, the question is whether it will terminate the strand or whether it will just cause a mismatch to happen that
Right.
will
That's right.
or like change the sequence. So
Yeah, this
this
could
is
be like introducing mutations or whatever. But again, we won't see that from Chymograph data. That's just
no,
like from what we know of the mechanisms of DNA replication, right? Like that's what you could predict from knowing this about the structure.
which is why it'd be interesting if there is a change in the chymograph because we wouldn't expect that. So let's see that data. I discovered PowerPoint transitions, so now I'm going to use them all the time.
Yeah, that was really dramatic. I was like, oh my God, the data's coming. Star Wars style.
Exactly.
Um,
So
it does. These are
yeah,
screwed
it
up.
does.
These are screwed up.
Yeah, the more you add, I mean, admittedly, you have to add a fair bit, and so it's now in the ratio of like 200 and 500,
Yep.
but it still has like an effect, not
Yeah.
so, on
Well,
the speed at least.
these aren't ratios. These are just the
Oh, right.
concentrations. Yeah. So
Okay.
I think this is under the 500 micromolar condition. So like these are going up to 1 if you're thinking about ratios. These are going to 1,
So
0 to 1.
yeah, this actually like slows down the RDP, so that's
Yes.
interesting because I wouldn't
Yeah.
necessarily have expected that.
Yeah, no termination, but yeah, a slowdown. And there is some backtracking still, right? We're also seeing an increase in in the probability of seeing backtracking. So again, I think to me this is making sense from we know that the polymerase is a proofreading polymerase, right? So if it sees at some point, like I'm sure it knows that it did something wrong, and it's just taking time to figure out like what's wrong. Like, does it need to like diffuse a little bit back and forth to like find the error and potentially excise it? Because again, the product length, or not again, the product length in these circumstances, they aren't changing that much, right? Like
Right?
even
Yeah.
at the 1-to-1 ratio, like those products are still, yeah, those are still pretty much full-length products. 946 versus 940 at the 0. Of course, that's within some margin of error. So
Yeah.
they do finish eventually. It's just that they pause a lot. It takes a longer time.
Yeah. I think that's because you wouldn't expect them to be a chain terminator because they still have the ability to. They've got the Lego brick connections between each other. Just the fact
Exactly.
that it slows everything down because for some reason it seems like maybe the RDRP knows it's made a mistake and is going back to check.
I think so. That's my suspicion with seeing this data. It's like it trips something in the RNA dependent RNA polymerase. It's like, what's going on here? And then eventually, like, right, like the way that thermodynamics is like, well, there's nothing stopping me from just completing the whole thing, so like continue on.
Right. And so yeah, they did this again with different forces just to confirm that it happens.
Yep.
Um,
They're still seeing that.
uh, yeah, we're still seeing the same effect. And now we've got like two candidates. We've got sulfosbuvir and
Yes.
3-DUP-UTP, which are similar to ATP. That's no wait. I mean
Oh, that's
I can't
probably a
remember which
bad.
one this
That's probably they're similar to UTP.
UTP. Yeah, that's
Yeah.
right.
Yeah.
Uracil, which is we usually
Uracil.
yeah.
So this sofosbuvir, oh my goodness, I hate saying drug names. This particular molecule, it's approved for HCV. So HCV is another RNA positive strand RNA virus. So like in a similar family, there. So the sense is like, oh, if it works for that, it might work for SARS-CoV-2. But of course, like their RNA-dependent polymerases may be quite different, right? So just because it works for one doesn't necessarily mean it'll work for the other. I think what's interesting is that people say that sofosbuvir is like sabuvir is a chain terminator, but it has that 3' OH, so it can do that chemistry. So the way in which people think it might terminate the chain, like, is different than just not being able to do the chemistry. Maybe the fluorine's a pretty big molecule, so maybe having it sit
Yeah.
right beside the OH there might like somehow mess up the catalytic site, so it does effectively prevent that chemistry from going.
Yeah, I mean, remembering my high school chemistry, I think it might like steal a bunch of electrons from the crowd. I think
Oh yes,
there's
that's another really good point. Yeah, it would make the OH less electropositive, right? More electronegative, potentially.
all I'm gonna say is it will change it. I am not like this is my high school chemistry. I don't know what it'll do. Maybe
No, no,
it'll
no.
do some difference. Is what.
No, I'm gonna dogpile on that. I think you're right. Like I think it, and I'm gonna claim that it's pulling the electrons away, so it's making it more positive. Because the
Step away
phosphates are
from my bad idea. Just
the phosphates are
I'm handing
negative.
over to you.
Yeah, I'll accept.
Yeah.
I'll accept responsibility. Yell at me in the comments if that's
Yeah.
incorrect.
Yep, another fine mess I'm walking away from.
Yeah, so this is interesting because this is something that's already approved, right? So like having some knowledge
Yeah.
about this, like could, yeah, maybe it should be working, but there's other reasons why it doesn't work. But actually, no, that's not what they find. It's not that great at slowing things down.
Yeah, there is
Yeah.
basically no difference in the behavior. So
Yeah.
it doesn't look like it's doing anything.
Yep.
So I guess it must be that's because SARS-CoV-2 has all these other activities that are different from the disease it's supposed to be working against, which I've forgotten completely. But
Yeah.
HCV.
HCV. Yeah, it could be that the backtracking or the proofreading is better, right? Like it's really effective at just like busting through this. Yeah. Anyways, it's not showing us anything at this particular concentration, but let's see what we see in our next figure.
Yeah, so now this is looking at
UTP.
DUTP,
So
which is lacking that Lego connector, right?
And this one is quite effective in terminating the chain. So we're seeing like a decrease in the product lengths that are being made. And also, I mean, consequently, a decrease in the replication time, but that's again because of the decrease in the product length. And yeah, it doesn't really do anything with the pausing as much. This is a strict chain terminator.
Yeah, so it behaves exactly. So the polymerase behaves exactly as it normally would. It would just end quicker because it would just be like, oh, we've come to the end of the chain. Well, time for something else to do.
So they'll now jack up the concentration or they'll lower the concentration of the UTP, or the good nucleotide, effectively increasing the proportion of the nucleotide analog. And at this level, they do see an effect with sofosbuvir.
Right. Yeah, they see like more chain termination events where the chain just like the RNA polymerase just gives up. So there is something happening with that fluoride that's doing something. I don't know what.
Yep. But you have to have like a higher concentration of it relative to the natural nucleosides around, which, you know, that casts severe doubts on this ability of this to be an effective antiviral in this circumstance, because like it's kind of crazy to imagine that you're gonna overwhelm your body with so much nucleoside analog that like it's greater than the concentration of regular nucleosides in your body.
I mean, we can only really compare to other things in this experiment because we don't know whether like Remdesivir suddenly won't be like overwhelmed with nuclear. So it's
yeah, that's true. Very true.
So I'm not going to write off because sofosbuvir probably has a family and they might be really upset with us. But I don't want to be just going, oh, sofosbuvir, they're terrible. We're not friends with sofosbuvir anymore. Like, hang on, like.
Yeah, at this level, it is way less effective than Remdesivir.
Yeah, way less effective, and it doesn't seem to have any changes into the behavior that we'd expect. But it does behave like a chain terminator, which tells us about how it works.
Yes, which again we also knew ahead of time. Right? Like the experiment is confirming these types of things for us.
Yeah, and lowering the concentration needs to be dramatic. So now we definitely know it's a chain terminator because nothing gets over 250, where it needs to reach that 1,000 mark, and they're just not getting there.
Yeah, well, a big part of I think the thrust of Figure 5 for the authors was just comparing the chain termination ability between DUTP and sofosbuvir, right? And like if something is lacking that OH, it is a very effective chain terminator, right? Not having the ability to do that chemistry is stopping. Having something that might like mess up that chemistry, like RNA-dependent polymerases can get over that unless you jack up the concentration, in which maybe there's just so much blockage from all those extra bulky groups that the enzyme is failing to figure out what to do.
Yeah, and so now we've got like the DTCTP, so it's a cytosine analog, right?
Yes. I think this is the final one they're going to look at in this paper. So DDCTP is like kind of cool. It naturally occurs in our cells. So this is like another line of thought, like an interesting line of thought. This could be part of like our natural antiviral immunity is just like low amounts of this DDHCTP in our bodies. And again, it looks very similar to a classic chain terminating analog, DCTP. And so we would expect this to be a chain terminator as well. But of course, it's naturally in our cells, and we get infected by SARS-CoV-2. So likely SARS-CoV-2 is going to be able to somehow overcome this. But let's see what the data has in store for us.
Yeah, let's zoom into this data, and now we're looking at DDHCTP, which looks like it does do some chain terminating, right?
Yes, yes, and it's increasing the backtrack ratio as well slightly, which is interesting to see. So it is, like SARS-CoV-2 knows something is going on, and the RNA-dependent RNA polymerase knows something's going on, trying to figure it out, but ultimately it's a minor effect as compared to what we've seen previously.
Yep, and now we've got 3D-CTP, which is the classic chain terminator.
Yeah.
And here we are seeing some, in comparison, like much more chain terminating is happening.
Yeah. I find that fascinating. To me, this almost speaks to like I think that this data is now trying to say something about the evolution of our RDRPs, right? That like, oh, actually, because there is a natural nucleoside analog inside of our cells, like the conformation or whatever of how these RNA-dependent RNA polymerases work somehow addresses that version. But can't address this. Like the change between this is kind of. I mean, I don't know what counts as small or big in the chemical world, but like they're very similar, and yet this one has a greater effect.
Yeah, the only real difference between the two structures is like this one like double bond in the corner. So in DDH there's a double bond, and then in 3DCP you've got that hydrogen there.
Right.
So I don't know what effect that's happening, but it seems in this case to be exerting like some kind of effect on how an RDPR. So that's.
But again,
really
I want to like, and I just want to tell people like focus on the like thinking about from the RNA-dependent RNA polymerase perspective, right? Like it ostensibly has been evolved in a context where DDHCTP exists, right? So like it has like maybe some way, right? Like it already accounted for this, right, in its design. But this DCTP, it's not accounted for, and so we see a stronger effect from it.
Right, that brings us to lowering the concentration of CTP and jacking up the CTP. And
so it works.
We do see,
if you can get more of this, it does something. Yeah.
Yeah, it does something. Yeah, and then if we look at, say, DCTP, again, same thing. It
way
just
more dramatic.
That effect.
Way more dramatic.
Yeah. And so in terms of chain termination levels, we can now compare all the things we've tested so far, apart from Remdesivir, I think. But yeah, you can see that the main chain terminators seem to all be working in sofosbuvir.
Yeah, they work at varying levels, right? And this is like a summary. Like I don't know. This graph is kind of like randomly placed here, but it's like it's a summary of like changing out the tweezer force for 25 piconewtons versus 35, just across all the last 3 that we looked at, except Remdezvir, and it doesn't have any effect. Yeah, I probably should have cut this slide.
And now we're taking it to real cells.
Yes,
So,
yes, yes.
yeah.
And again,
So,
this data already existed, right? So like we know, like other people have done these studies, but it is nice to see it also in the hands of this group. It can tell us something like we know that they're using the right stocks, right? Like they're not like out there with some weird reagent. And again, it builds up that story, right? To connect the low affinity, right, of sofosbuvir to SARS-CoV-2 RNA-dependent RNA polymerase. Yeah, that low affinity to an inability to be able to suppress SARS-CoV-2 infection in cell culture. And similarly, right, the low ability for DDH-CTP to, I guess, even terminate chains in this sense, yeah, is being demonstrated here.
yeah, and it shows Remdesivir is much better than the other. And I think that also demonstrates that how the tweezers as a technique to find antivirals can be quite useful. Because this gives
Yeah,
us
yeah.
like the threshold of what we want to look for.
Right, like in this case, right? In this case, we already had this information, right? We knew the relative effects of these different things. Well, maybe not DDHCTP, right? But again, yeah, the authors, they're really concerned about like showing the utility of their tool in this particular paper. So yeah, I agree with you. In some ways, imagine though, right? If we didn't know this information, and so instead of doing cell tests, we just did these tweezer tests and we were able to test a whole bunch of different antiretrovirals, right? We could then predict, right, the types of traces that would produce good candidates to then move into cell culture. Because cell culture is more expensive than doing things, right, in biochemistry style in a flow cell. And so, yeah, that's a really good point. This is like we're relevant, our techniques are relevant.
And so, like, so
Oh,
based on
yes.
this, I think, yeah.
They
Sorry.
they kind of have like a tangential follow-up where they're like, ooh, you know, like DDH-CTP, like that could be maybe the reason why, right? It's not doing a good job is because NSP14, the proofreading enzyme, again, as I was saying, has evolved under the circumstances where, yeah, it just like lops it out and like business as usual continues. So if they remove that ability, then maybe they will see they'll see an effect, right? But unfortunately, in the process of removing that ability, they also just removed the ability for SARS-CoV-2 to go in and infect cells properly. So they can't actually follow up with that hypothesis.
Yeah, that's unfortunate. So, yeah, no. So, yeah, I think that brings us to towards to the last figure in this paper.
Yes, just the summary of what they found, right? They had tweezers, they used tweezers. And they were able to come to some understanding of what the mechanism of action. First of all, I mean, we knew the mechanism of action for some of these things. I guess it is surprising, right? People thought that Remdesivir was like a chain terminator, but
Right.
actually, like we're seeing, like actually it induces this like backtrack ability that then maybe leads to some chain termination. So it's a weird one. And again, we have other literature to sort of flesh out the story of Remdesivir. But yeah, this is the data in summary that they pulled out in this paper. Ooh.
Yeah, and that brings us to the end of our deep dive. So, yeah, what are our final thoughts about this paper?
Cool. I like again like this. I think my thought with seeing the cell culture and how they're arguing for relevance of this technique is what I'm also excited about for them is that they have what seems like a great way of getting at yeah physiologic or yeah a biochemical reading that has a strong correlation, right, with how we understand the physiologic effect would be, and thus is poised to be a good way to screen through many different types of antiretrovirals, yeah, in relation to RNA-dependent RNA polymerases.
Yeah, and I think the interesting thing about like understanding the behavior of these polymerases at this level is all
Mhm.
like because I mean thinking in terms of like the like if we want to like engineer our own polymerases and finding ways to test how they work, or because I think like this is a nice little tool to pick out like how often a behavior happens. And
Yeah.
quite a lot of this paper isn't just about like the technique of but it's also about the data analysis, which again is using those probability curves, which is a completely, well, it's a new thing for me, but probably isn't very new for people working in the field at the moment. But
Yes.
it's
Yes. I mean, it's in some ways it's like a very in-depth view into like where enzymology has went, right? Like Michaelis Menten, right? Like these graphs of like, oh, you add more substrate as the rate increases and then it plateaus at some point. Like this is like now like we're talking about like 4 different Michaelis Menten graphs because the enzyme can like based on what we've seen of its behavior, right, through across many different single molecule experiments, its behavior can be put into 4 different categories, right? So like there are things happen, like these are big proteins, like when something binds to it, like who knows, like, I mean, we can't say like what the shifts are like on other parts of that protein. And this technique is giving us like a rare glimpse, right, into that level of resolution to be able to distinguish like potentially right 4 modes, underlying modes of the way that this enzyme works. Still, again, it's a model for data, right? Like those 4 modes, like they're being inferred, and we have to trust them because like we're not good statisticians here, right? Like to be able to vouch for what they've done, but like. Yeah, 4 different modes that are, you know, rigorously statistically supported, right, as having existence based on this data.
yeah, yeah, that's yeah, because like often when you get data like this in like back when I was like in undergrad, it would basically write off that kind of behavior as like it ended up in the standard deviation. But now we're in the point of like single molecule kinetics, where you can just look at how each molecule is behaving individually at a time. That's really like a new thing for me.
Yeah,
And
yeah.
so yeah, and also it's a neat technique that again high throughput, so you can do lots and lots of tests at the same time. Very interesting, and also like yeah, like what they did here, it can be done at a much larger scale with, because I think like we've seen a bunch of like papers where they just do RNG on like structures for antivirals and then just throw them at something,
Yeah.
and this seems like a much like more rapid and informative technique about how they work.
Yes. Yes.
Because
Yes.
I can imagine this with like any like virus polymerase effectively, and with
Yeah, it's very
so
extensible, right? Like if we want to figure out anything, and then again, we also have to think that the tweezer technique, this is just one way that tweezers can be used, right, to set up to study a specific enzyme. But really, a lot of things may be able, like people have studied just like DNA, like regular RNA polymerases, DNA-dependent RNA polymerases using this method, right? So like it's not even just RNA-dependent RNA polymerases, that anything that is grabbing onto a piece of DNA can be studied through this mechanism. You can get like a higher resolution basically on how those enzymes work. I just want to say,
yeah, so
Minref,
yeah.
I'm sorry we missed your comment when we were talking, but yeah, we've been saying chymograph.
Yeah, Kymograph.
Yeah,
Yeah,
yeah.
so
The traces here, yeah, the traces in this paper have been, yeah, distance over time. And so, yeah, the distance of these little, like, yeah, balls.
yeah,
That was like not explanatory at all. The distance of these balls, the distance of these balls in the focal plane as correlated to right the extension of nucleic acid.
I'm just gonna keep making you explain this because it's so funny when you just go
Balls.
but it's just, but yeah, so yeah, it's
Yeah,
yeah,
that's my
we're
takeaway.
measuring the motion of
Yeah,
things.
this is very biochemistry, right? Like I
Yeah,
actually like, but again, microbiology, like we're here to understand the organisms, right? And all the varied ways in which those organisms come to impact our lives. SARS-CoV-2 has been one tiny little bugger that has been doing a lot to us as a society, and these authors are getting at understanding that particular bug through the lens of its RNA-dependent RNA polymerase, a very druggable target, and this technique I think is going to give us be able to screen through candidates for that druggable target a lot faster. As well as if you guys are creative assay developers, you could maybe think of 1,000 ways in which these tweezers could potentially give us more insight into enzyme kinetics.
Yeah, I think that's a great way to end the show.
All right. So join us next week where we're going to do a deep dive, do a news overview on some of the coolest microbiology papers that we've seen in the past few days.
And we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Science is about about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach out to us over Twitter or Mastodon with the hashtag #MicroTWJC. We both believe that peer review is a process that we can all be a part of, and we hope that you've had a good time listening to us ramble on about microbiology today. If you think you have something to add or found something unclear, just let us know in the comments.
Oh, did you just mention Mastodon? So can you tell us a bit more about our new Mastodon
Oh,
account?
yes. Yeah, I'll try to figure out a better hashtag for us over there, but we're also on Mastodon. It's just a different social media network. I'm particularly wedded to it because, we're dodging a little bit of the Twitter craziness and having having admins that sort of that they're better poised to control the discussion there. But again, it's just another surface in which people can reach out. If you want to dive into the federated, the Fediverse, as people call it, we're happy to be your entry point into it.
Yeah, thank you. It's been a pleasure chatting with you.
Same here, Fuzz.
Tune in next week for more microbiology content. Bye.